Hello student, welcome in this Rose online project. Rose online project is conducted by Rare Shikshan Sanstha, Satara, Sadguru Gadge Maharaj College, Karad. Myself is Pawar SP from SGM College, Karad. My subject is biology. So today we will again discuss the topic that is molecular basis of inheritance. Today's lecture number 6. In previous lecture, we can understand what is protein synthesis, which step involved in protein synthesis, and what is the meaning of the transcription. Transcription, that is the formation of the mRNA from DNA. And then we also learn gene regulation. Okay, so today in this lecture we learn the important term. This term is known as a translation. Okay, translation, the process in which the sequence of codon on the mRNA strand is used, that is read, and accordingly the amino acid are joined to each other to form the polypeptide chain for formation of the protein. Then translation is the mechanism in which codon of mRNA are translated and specific amino acid in a sequence from a polynucleotide chain on the ribosome. That is, formation of the mRNA from DNA. This process is known as a transcription, and formation of the polypeptide chain, that is synthesis of the protein from mRNA. This process is known as a translation. So protein synthesis involves two processes. First process, it is known as a transcription, and second process, it is known as a translation. Transcription and translation are the two major processes of the central dogma. So here watch the diagrammatic representation of two processes, that is transcription and translation. Formation of the mRNA from DNA. This process is known as a transcription, while synthesis of the protein from RNA, this process is known as a translation. Transcription process in eukaryotic cell take place in nucleus, and translation process take place in eukaryotic cell in cytoplasm. Translation occur in prokaryotic cell in cytoplasm. So translation meaning the formation or the synthesis of the protein from mRNA. This process is known as a translation. Now see, for the process of the translation, which component are the required? That is amino acid, mRNA, tRNA, ribosomes, ATP, Mg ion, enzymes, elongation, translocation, and release factor. Then amino acid form raw material for protein synthesis. About 20 types of the amino acid are known to form a protein. They are available in cytoplasm. DNA, this nucleic acid, control the protein synthesis having amino acid in specific sequence. This control is possible through the transcription of mRNA. Genetic code is specific for particular amino acid. So RNA serve as an intermediate molecule between DNA and protein. Ribosome serve as a site for protein synthesis. Each ribosome consists large subunit and small subunit. These subunit occur separately in cytoplasm. Only during protein synthesis these subunits get associated together due to the presence of the Mg++ ion, that is magnesium ion. This phenomenon is known as a association. So ribosomes play important role in a protein synthesis. So ribosomes are called as a protein factories. 70S type of the ribosome present in the prokaryotic cell and 80S type of the ribosome present in the eukaryotic cell. And both type of the ribosome consist of small subunit as well as a larger subunit. They present separately in cytoplasm. But during the process of the protein synthesis, these subunit associated due to the magnesium ion. So this process is known as a association. And ribosome has only one binding site for the mRNA and 3 binding site for the tRNA for the synthesis of the protein, that is for the process of the translation. DNA molecule is required, non-genetic type of the RNA, that is mRNA, tRNA and rRNA. These non-genetic RNA also required, and they also use source of energy, that is ATP and GTP. So ribosome consists of one binding site for the mRNA and 3 binding site for the tRNA. And then they consist P site, that is peptidyl tRNA site, A site, that is amino acyl tRNA site, E site, that is a exit site. Only first tRNA amino acid complex directly enter the P site of the ribosome. In eukaryotes, a groove is present between the two subunits of the ribosome, that is smaller subunit and larger subunit, and it protects the polypeptide chain from the action of a cellular enzyme. They also protect mRNA from the action of the nucleus. So here see the diagram of the ribosome. Ribosomes are known as a protein factories of cell. Ribosome consists of two parts, smaller subunit and larger subunit. And ribosome consists of one binding site for the mRNA and 3 binding site for the tRNA. 3 binding site, that is E site, P site and A site. E site it is known as a exit site, P site it is known as a peptidyl site, and A site it is known as a amino acyl tRNA site. So this is the structure of the ribosome. It is small ribosomal subunit. It is larger ribosomal subunit. It consists of one binding site for the mRNA and 3 binding site for the tRNA, that is E site, P site and A site. Okay, now see the mechanism of protein synthesis. Mechanism of protein synthesis consists of essential step, that is activation of the amino acid and formation of the tRNA complex. And this activation of amino acid takes place in the presence of special enzyme. This special enzyme is known as a amino acyl tRNA synthesis. So activation of amino acid, that is formation of the tRNA complex, take place in the presence of the enzyme amino acyl tRNA synthetase. The amino acid molecule activated, then each amino acid, activation of amino acid, that is amino acid attached to specific tRNA molecule at 3' or 3' end, that is a CCA end, to form AA, amino acyl tRNA complex. AA tRNA complex means amino acyl tRNA complex and activation of amino acid. This process use the source of energy, that is ATP. What is the long form of the ATP? Adenosine triphosphate. So activation of amino acid, that is formation of the tRNA complex. This process is also known as a charging, and this process takes place in the presence of the amino acyl tRNA synthase. They also use the source of energy, that is ATP, and amino acid attached to the specific tRNA molecule at the 3' end, and there is the formation of the amino acyl tRNA complex. So it is the important process in the mechanism of the protein synthesis. Then the second step of the translation, that is the formation of the polypeptide chain. First step, that is the activation of the amino acid, and second step, that is the formation of the polypeptide chain. But formation of the polypeptide chain again consists of 3 steps. Which 3 steps? First step, it is known as a initiation. Second step, it is known as a elongation, and third step, it is known as a termination. So formation of the polypeptide chain involve initiation process, involve elongation process, involve termination process. So now see the first process, that is initiation. Initiation, it begins with the formation of the initiation complex. It requires mRNA having codon. Codons present on the mRNA and anticodons are present on the tRNA. Okay, so for initiation process require the mRNA having codon for the formation of polypeptide chain, and the two subunit of the ribosome, that is 30S and 50S ribosome, and initiation complex, that is AAtRNA complex, and source of energy, that is ATP and GTP is used, adenosine triphosphate and guanosine triphosphate. So in initiation process, mRNA having codon for the formation of the polypeptide chain and two subunit of the ribosome, that is 30S and 50S. Initiation AAtRNA complex, and they also use ATP and GTP as a source of energy. This process also required initiation factor. So now see, in prokaryotes, the first AAtRNA complex has amino acid, that is formyl methionine. This is the important point for MCQ. Prokaryote, first AAtRNA complex has amino acid, that is formyl methionine, and in eukaryotic it is known as methionine. In eukaryote methionine, and in prokaryote F-methionine, that is a formyl methionine. The process starts with the binding of mRNA in smaller subunit of 30S on the ribosome. AUG is known as a start codon, which is positioned properly. Then AAtRNA complex, that is formyl methionine tRNA, then get attached to the start codon, that is AUG. This is done with the help of the activation of the UAC of tRNA. Small and large subunit of the ribosome join and form a 70S ribosome that require magnesium ion, and initiated charger tRNA occupies the P site of the ribosome and A site is vacant. Now, at the P site of the smaller subunit of the ribosome, this is done by the activation of the UAC of tRNA. Then small and large subunit of the ribosome join and form a 70S ribosome. The ribosome has 3 sites. मगाशी सांगितल्याप्रमाणे 3 साइट कोणत्या? Amino acyl site, that is A site, peptidyl site, that is a P site, and exit site, it is known as a E site. After translation over, the empty tRNA leaves from the E site and the AA tRNA complex binds at the P site directly, and other incoming tRNA complex get attached at the A site, and then they are shifted to the P site, and polypeptide chain which is being formed is released from the P site. So next process in the formation of the polypeptide chain, that is elongation. After initiation, the process of elongation start. Okay, this is done by the formation of peptide linkage between the successive amino acid molecule. The elongation process catalyzed by the enzyme peptidyl transferase. This is the important point. Activation amino acid synthetase enzyme is necessary, and for the elongation enzyme peptidyl transferase is a necessary, and each tRNA complex bring a specific amino acid due to the complementary nature of anticodon. Elongation activity also involve elongation factor. Addition of the amino acid occur in 3 steps. Codon recognition, that is amino acyl tRNA molecule enter the ribosome at the H site, and anticodon bind with the codon by the hydrogen bond. Amino acid on the first initiated, that is tRNA at the P site, and amino acid on tRNA at A site joined by the peptide bond. Here enzyme ribozyme act as a catalyst, and translocation, that is the tRNA at the A site carrying a dipeptide at A site move to the P site. This process is known as a translocation. Then both the subunit of the ribosome along in relation to tRNA and mRNA. tRNA carrying dipeptide now get positioned at the P site of the ribosome and making A site vacant at this site. The next charged tRNA molecule carrying amino acid will be received. During this process, first uncharged tRNA is discharged from the E site. This process is repeated as amino acid are joined to polypeptide. It takes then 0.1 second for the formation of the peptide bond. The third charged tRNA with its amino acid arrives at the A site of the ribosome. Anticodon which are present on the tRNA and codon which are present on the mRNA bind by hydrogen bond and polypeptide bond formed. Okay, and second tRNA then discharged from the P site to the E site and leaves the ribosome. So the event like arrival of the tRNA amino acid molecule, formation of the polypeptide bond, ribosomal translocation, and removal of the previous tRNA are repeated. As ribosome move over the mRNA, all the codon on mRNA are exposed one by one for the translocation. So then the last term, that is a termination and release of factor. At the end of the mRNA, there is a stop codon, that is UAA, UAG, UGA. It is exposed at the A site. It is not read and joined by anticodon of tRNA. The release factor bind to stop codon. thereby terminating the translation process. The polypeptide is now released in the cytoplasm. Two subunits of the ribosome then dissociate and last tRNA is set free in the cytoplasm. mRNA also have some additional sequences that are not translated and are referred as untranslated region UTR. What is the meaning of UTR? Untranslated region. And untranslated region are present at both, that is a 5' end, 5' end that is a before start codon, and at the 3' end after the stop codon, and mRNA released in the cytoplasm. So this is the diagrammatic representation of the translation process. This is the structure of the mRNA. mRNA consists 5' end and 3' end. At the 5' end starting codon, that is AUG present. At the 3' end stop codon, that is AUAA codon is present. Ribosome consists two subunit, smaller subunit and larger subunit. Larger subunit consists 3 site, E site, P site and A site, and tRNA bring anticodon. These anticodons are complementary to codons which are present on the mRNA, and these anticodon and codons are binding together by the hydrogen bond. In this way, at the 5' end, translation process start. Then for the polypeptide chain formation take place, and at the 3' end the termination process take place. Now see here again the diagram mechanism of the translation. Mechanism of translation involve 3 steps: initiation, elongation and termination. So here watch initiation, that is at the 5' end. Initiation process start in the elongation process, formation of the polypeptide chain former, and termination at the 3' end. Termination process take place. Okay, in this way the translation process take place. Now see the regulation of the gene expression. What is regulation of gene expression? It is multiple process by which gene is regulated and its product is synthesized. Thus, gene expression result in the formation of a polypeptide. Gene expression process is regulated at different level. Okay, in eukaryotes, the regulation can be at different level like transcriptional level, that is formation of the primary transcriptor; processing level, that is regulation of the splicing; transport of the mRNA from nucleus to the cytoplasm; translation level. Okay, for example, lactose get converted into galactose and glucose in presence of the beta-galactosidase enzyme. If E. coli bacteria do not have lactose in the surrounding medium as a source of energy, then the enzyme beta-galactosidase is not involved. So it is the metabolic or physiological or environmental condition regulate expression of a gene. Certain bacteria like E. coli adapt to their chemical environment by synthesizing certain enzyme depending upon the substrate present. Such adaptive enzyme is known as a inducible enzyme. A set of gene will be switched on when there is the necessity to metabolize a new substrate. This phenomenon is called induction, and small molecule responsible for this is known as a inducer. It is a positive control. So thank you watching this lecture.